Hey everyone, hope you're all doing well and everyone's healthy and happy. We're going to start genetics, which is my favorite part of living environment, and it's kind of sad that we can't be together to do all the fun experiments that I usually like to do with this. But I might be able to find some cool stuff for you guys to do at home. Um, yeah, all right. So when we talk about nucleic acids or genetics, we always talk about DNA and RNA. So DNA is, most of you guys know, is the main nucleic acid, and RNA is equally as important, but we haven't really talked about it yet. Okay. So today we're going to talk about the nucleic acid. Okay. Those are one of our four major molecules. We've done a lot already about proteins, lipids, and carbohydrates, but now we're going to talk about nucleic acids for a while. Okay. So there's different bases. The four letters you guys might know. We'll get to that in a minute. Okay, and where we can find DNA, which I'm sure most of you know already, is the nucleus. Okay, so there are building blocks to DNA and RNA. Okay, so there's two types of acids that we talk about. Okay, so DNA stands for deoxyribose nucleic acid, and RNA is ribonucleic acid. And if you notice, the only difference in these long names is this part here. Okay, if we take out the deoxy part, then it's just ribonucleic acid. So what this means is that compared to RNA, DNA is down one oxygen molecule. So these acids are made up of three different parts. Okay, a phosphate group, which on the periodic table is letter P, bonded with other elements. The nitrogenous base, nitrogen, we see in the name, so that's N. And our simple sugars, which, if you guys remember, is just C6H12O6. Okay, now it's not always C6H12O6, but it is a simple sugar, and the simple sugar we know is glucose, which is this one here. So they all make up a nucleic acid, whether it be DNA and RNA, but the base, okay, becomes a major difference. So for DNA, okay, our bases are A, T, G, and C. Okay, and you don't have to worry about the larger names of these. Okay, but where it says what are the four bases for DNA? Okay, that's our A, T, C, and G. And we'll look at the longer names in a minute. But when we think about a nucleotide, okay, it's helpful to kind of think of it like a house, okay, or a nice house, right? Because I don't have a pool or a detached garage or a house that looks like this. Anyway, so the simple sugar in this case, all right, is the house. Okay, it's the main part. It's in the middle. Okay, and if you look on either side of the house, you can find the nitrogen base, okay, which are these guys here, or you'll have on the other side the phosphate pool. Okay, P for pool, and it usually takes on a circular shapes in most diagrams. So we'll look at that in a minute. So here are the long forms of them: adenine, guanine, cytosine, and thymine. But again, what you should be concerned with is the A, G, C, and T. When we talk about RNA, thymine, so T, is replaced with the U. Okay, and that's for uracil. Oops, Gilbert. Yep. Okay. So uracil is just what it acts like the T, but it becomes important later on we'll talk about when we get to the processes that DNA and RNA are used in. Okay, so the bases make up the rungs of the DNA ladder, so to speak. What are the rungs of the ladder? So that's these guys here, these long pieces. These are your bases. Okay, so in between, well, on the other side of this would be another strand. Okay, so here's our 4 bases, here's our pool, and here's our house. Okay, and we'll look at a non-spinning picture I can draw in a moment. So, but these form pairs, okay, and then they lock together. So adenine, or A, always pairs with thymine, T, in DNA. Okay, and that means C, cytosine, must pair with G. Okay, C won't pair with T, G won't pair with A. That's a mutation. We'll talk about mutations too in this unit. Okay, so this is called base pairing. So if I ask you what's the base pair for adenine, you would say thymine, or A for T. Okay, when these bond together, that's what makes up this DNA ladder. Okay, that's how they're held together. And of course, you could pause the video if you need to keep taking notes. But if I can draw using my Picasso-like art skills here. Okay, so this here represents our sugar, and phosphate. I don't know how people do this. Okay, and then our bases would be in the middle here. And these are your base pairs. Okay, so here's the non-spinning pictures. So this is essentially what I just draw, right? This was the single line that I drew last time here. So here's our sugar, here's our phosphate, and you can see the P in the middle here. The hydrogen bond, okay, that's just the type of bond that holds these together. Just know what pairs with what though, so A with T. Okay, and this is 3 different ways to look at it, right? So this is, you might have seen this kind of picture, and this is like with all the letters and stuff taken out, what it would look like if we were to take it out and put under a microscope and then filled in the empty spaces with bubbles. Okay, so how do you remember this? I like to think, like, where would I want to be right now, right? Actually, probably one of the best social distancing places would be this, right? At the Grand Canyon. A and T go, and G and C go together, right? If you guys can think of any other ways to remember it, let me know. Okay, maybe I'll post them on Schoology for all to see. Okay, another one is good couple, or good couples are always together. Okay, or good cheese, always tasty. Good cars, always trains. Okay, in most types of cells, DNA is present inside of the nucleus. Okay, so imagine like you bought a dresser from IKEA, and then you have all these pieces, which are basically like your DNAs, your RNAs, okay, amino acids we'll talk about in a little bit. And the instruction booklet is the DNA itself. Okay, that's going to tell you how to build whatever it is. So height, hair color, okay. These are all things that are determined by DNA, okay, in genes that supply the instructions on how to do it. So at some point, DNA is read by an enzyme that translate the DNA language into RNA language. And the way that the DNA is set up will determine which RNA molecule is made and then brought to make proteins. Okay, and this is what's called like the central dogma of biology. Okay, it's like the basic rule. It's how everything is made from the body. Okay, so DNA to RNA, and then to our protein product. Okay, so segments of DNA are transcribed. We'll get more into this. This is just an overview, okay, by an enzyme, which we remember is a protein. Okay, and that's made to mRNA. There's different types of RNA molecules, okay, but we'll look at why this is an M probably within the next few days. So this mRNA carries a message and is translated into proteins by ribosomes. Okay, we remember, right? Ribosomes like to eat ribs, so they make proteins because ribs are made out of proteins, or however else you want to remember it. Okay, in the cytoplasm. Told you guys organelles would come back to haunt you. Not haunt you, but you need to know what they are so you can better understand this. So if you guys have any questions about the organelles mentioned here, please let me know and I can answer them. Okay, so the ribosomes can be found in two places, right? They could be just hanging around the cytoplasm or bound to the endoplasmic reticulum. Okay, and when these are bound together, this makes up the rough ER, not the smooth ER. Okay, and they're made by a third type of RNA, okay, called rRNA. That just means ribosomal RNA, okay? So ribosomes are made up of rRNA. They have enzymatic properties because they speed things up and they make proteins, but they're not really proteins. There's a more complex name for that, but we don't have to get into that. So if you see enzymatic property, that's just because it speeds up, okay? And the synthesis of protein, okay, is our reaction, right? We said ribosomes speed up reactions. They're catalysts. So when we say synthesis of proteins, that's our chemical reactions. And for a refresher, right, we have ribosomes here, and they can also be found, it's kind of hard to see in this picture, but they are on the wall of the ER here. Okay, and this is a more close-up picture. This is the RNA going into the ribosome. Okay, now this is a very confusing picture, I know that, but I just wanted to give you an overview of what we're going to be talking about next. Okay, so here's our DNA. Okay, that enzyme I was telling you about that reads the DNA and turns it into RNA is called an RNA polymerase. Okay, so this is 1, 2. Here's our RNA that is made from that, the transcription. Okay. This then moves out into the cytoplasm or the ER. Okay, and here's our ribosome. Okay, and this is where our polypeptide chain, remember that from the beginning of the year? Right, we said that amino acids put together can be blanking here. Oh, monopeptides, dipeptides, or polypeptides. So here's our amino acids that are brought in. Okay, so they go here and they build our protein chain. So the main differences between RNA and DNA. Okay, RNA is single-stranded. DNA is double-stranded. If it helps you remember, right, there's a D in DNA. You can think of that for double-stranded. RNA has uracil, where DNA has thymine. Like I said before, the sugars may be a little different. That's why deoxyribose and ribose are two sugars, right? So this means no oxygen, or one less oxygen molecule. Okay. So that's it for today. Kind of take it easy. If you guys have any questions, let me know. Don't forget you check your emails every day. Important information from me or Dr. Faccio or your other teachers. Yep. So see you guys. Oh no, but I'll be making more videos. If you want to ask me anything, please email me.